situation. But hopefully those of you who are much more facile with deep learning or other more complex modeling strategies will find at least some of the ideas worth considering from a more conceptual standpoint. So with that, I think that's the preamble. If you, Fabio, want to take over or start the recording or switch officially to talk mode.
No, good. You can start right now. Thank you.
Okay, you're very welcome. Okay, so assuming the recording is on, welcome again, and thank you for your interest in this presentation. I want to tell you a story that stretches over the course of 10 years, starting with maybe a simple case study, and then going to more recent methodological work of the lab. The presentation covers many topics somewhat briefly and perhaps superficially, but hopefully manages to give you a sense for what some of the challenges are in trying to think about using multiple studies to train statistical or AI models that are used for patient prognosis, patient classifications in health applications. Always like to start with disclosures, although none of the analyses that I described here involved any of the patents or license projects or any of the companies that I'm lucky enough to be involved in. So I think I can go over this fairly quickly. The only one that is remotely conceivably connected is this initial patent for Postin for debulking in ovarian cancer, although I think that the MGH let that patent expire, so maybe it's even time that I take that out of this page. As a frame of reference, I will use a lot of terms like reproducibility and replicability, particularly replicability, and I want to define it the way the statisticians do. I know there may be many computer scientists here, but I want to follow the National Academy of Sciences report on these two topics that came out in 2019. Recommended reading for anybody who is interested in this, and define replicability as the extent to which the result of a study are also found in subsequent similar enough studies, and reproducibility as the ability of other investigators to go in and decipher exactly what was done and in the analysis of a specific study, whether it be in the lab or with the code or anything else. So reproducibility is a property of a single study. Replicability is a property of a collection of studies. I know that sometimes in computer science, somewhat ironically, these two terms have evolved to have the exact opposite meaning. Interestingly, the National Academy of Sciences report, as good as it is, focuses very much on results defined in terms of hypothesis testing, but not so much in terms of predictions. And the word hypothesis test appears, the phrase hypothesis test appears 27 times, while machine learning only appears 3 times, and there is at that point, I don't think, any mention of AI. So I think replicability in the context of prediction is something that deserves more attention, and that's what the focus of the lab has been, and part of the reason why I'm giving you a talk that is a broad overview, as opposed to drilling into any of the specific contributions that we try to propose, is that I hope you catch the bug of thinking about the replicability angle when you do your machine learning and AI work. So just a bit of a quick roadmap. I'm going to start with telling you a little bit about a comprehensive review of study that are relevant for the joint training and validation of prognostic algorithms. In the specifics is algorithms for prognosticating survival of ovarian cancer patients using gene expression data. I think I'll skip the digression on meta-analytic training on signatures because I realize that we're already at 10:47, and I'll tell you how we use those to motivate realistic multi-study simulations to try to understand the trade-off between performance within study and performance across, and then move on from there to some novel learning architectures that we have been developing in the lab that are very generic ways to increase replicability across studies for just about any algorithm that you can think of. Finally, if I have time, I'll tell you a little bit about a more sort of conceptual slash philosophical slash decision theoretic hobby of mine, which is to try to figure out whether in the context of prediction, we can define replicability from a perspective that is motivated by decision theory. So I hope to tell you a little bit about that. So the starting point of this is about 10 years ago, and it is a meta-analysis that we did to try to evaluate, based on all the evidence available in the literature, the quality of prognostic models for ovarian cancer survival based on gene expression. And we did this sort of parallel slalom type of approach where we did a literature review, two literature review in parallel on the prognostic models that had been published and the curated databases that were suitable for the evaluation of these models. And so there, I will not bore you with the inclusion criteria and with the details of the implementation, but at the end of the day, we ended up with 14 prediction models that we could re-implement and entirely recode. We generated hundreds of pages of documentation and some packages to reproduce some of those models as well. But we had identified as early as 2012, I think, about 14 alternative models. And we identified also 10 independent data sets that contain the information that could be used to evaluate those models in terms of all the features, defining as gene expression measurements defined in a similar enough way, and as well as the phenotype of interest, and they were applicable to the type of cancer more specifically that we were interested in, which is essentially serous ovarian cancer. So part of that effort was to come up with database architecture for bioconductors that would be friendly to this type of effort, and we call that that first data set curated ovarian data. But after that, there have been several others that have been created under similar lines by ourselves and others. So you'll find in bioconductor things like curated breast data, curated bladder data, and a few others. And those, even though some of the gene expression data that is in there might be a little bit outdated from a purely computational biology standpoint, they make for a great didactical platform, and they're still quite interesting from a standpoint of methodological exploration. A lot of the points that I'm making in this paper are based on gene expression. Periodically, reviewers ask us, well, gene expression as measured by microarrays is not super interesting. We do it because there are many studies, and we can evaluate the sources of variation in a more systematic way. But a lot of the conclusions scale up perfectly to next generation sequencing, and we'll see what happens with the third generations. But I think that many of the statistical lessons remain, and for reasons that maybe we can discuss in more detail as we go along. So back to the 10 prediction models, and with 14 prediction models in 10 data sets, one simple thing that we can do is to evaluate the performance of each of the data set of the models on each of the data sets. And by performance here, this is a survival model, and the performance is measured by looking at the score produced by each model. It's a prognostic score that captures the severity of the patient's situation, and to put everything on the same footing, we use that score. However, it may have been determined. It could be a complicated score coming out of a neural network, or it could be a simple logistic regression linear term. And we plug it into a Cox proportional hazards model to get a measure of the hazard ratio. The stronger the hazard ratio, the more predictive the score. And we generate this matrix of these hazard ratios for each of the 14 models on the 10 data sets. We blacked out the studies that were used for training, and we also put in a gray box the studies that were reported as validation studies in the original publication to try to investigate the gap between within study and across study performance, and to also potentially assess biases in the literature driven by the fact that people were lucky or maybe somewhat strategic in choosing what data sets to validate on. There are many lessons from this matrix that we could dwell on, but the one that I want to emphasize the most, and where we will that motivated a lot of the subsequent work, is the gap between within study validation and cross study validation. It's very rare for a study to do as well in data sets that were not used as part of the training, and so I mean that's sort of a well-known fact. Well, before this study, this was maybe has the added flavor of documenting this in a way that is comprehensive with respect to both the algorithm in the literature at that time and the data sets available at the time. The next comment I want to make about this project is about whether or not these predictors rank patients similarly. And what I'm showing you on the left is a matrix that has, for the 14 rows are the predictors, and the columns are each individual patients in the merged data set comprising every study that we analyze. And you can see that there is some correlation, but the correlation is not very strong, which always suggested to me that there was opportunities for some serious ensembling, but standard ensembling strategies here didn't work for quite a while, and it took us about 4 or 5 years to figure out something that would give us a fair amount of improvement. The other piece of this figure on the right-hand side is a little bit of a digression, but I'll just tell you what it is as you're staring at it. It looks at the overlap in terms of the actual features that the various algorithms comprise, and the overlap is quite small in terms of the number of genes. The Jacquard index you see there is about 2% in the best case scenarios. But one needs to keep in mind that in the gene expression world, there are many redundant ways of capturing similar biological processes, and so this is reflected in the fact that the correlation increases quite a bit if you look at the gene sets where the features belong to the correlations themselves. Okay, so that's by the way of background and initial sort of inspiring application. I want to now move to the more methodological work and tell you first about a strategy that we've been following a lot for doing simulation study and experimental work, which is to leverage this type of comprehensive collection of studies to generate in silico data that is as faithful as possible to the real data. Not just one real data set, but the entire collection of data sets that we find in the literature about a specific problem. This is a lot of work, and you can only afford to do it once in a while. But there are now resources like this around, and you can often have the ability to do a sort of a bird's eye view of a field by getting a collection of data in this way. And I think that this is really important in terms of understanding the stability and replicability of algorithms that one hopes to deploy across a range of clinical or health-related or computational biology-related situations. So one of the questions that one can ask by generating in silico data that is very faithful to a large collection of empirical studies is, what are the sources of study-to-study variation? Because in silico you can control them. And this is a paper that we put in Biostatistics in 2018 where we do exactly that. The box plot represents the C index over a range of different generated data sets. The black and white are for the microarrays, and the color one are for RNA-seq. And the comparison is between CV, which is cross-validation within study, and CSV, which is cross-study validation. And you see at the left in the baseline case, which corresponds to the most realistic scenario in terms of matching the real data, that there's a systematic gap between the cross-validation and the cross-study validation. And so the question is, what do we need to do to remove this gap? We tried there are many things analyzed here. One is rebalancing the covariates, for example, and in this particular setting, given the covariates available, clinical covariate. So the features here are gene expression features, and the covariate represent more clinical type of features like stage, age, and things of that sort. And so balancing those did not particularly help with that. We tried to do some gene filtering with unsupervised methodology to try to understand the replicability of the genes without reference to any specific phenotype. Those tend to be somewhat restrictive. They do level out the playing field to some extent, but they also decrease the predictive performance a little bit, also cross study. We try to equalize the baseline hazard function, thinking that some studies may have more sick patients and others less sick, and that might be a driver. It is not in this particular case. And then we finally equalize what is here described as the model, which describes the relationship between the features and the outcome. So we essentially took out what computer scientists sometimes call the concept shift and made the relationship between features and outcome the same, and that's what really gets you to remove the gap between cross-validation and cross-study validation. Where is the concept shift coming in data in this type of data where you're using molecular features to try to predict health outcomes going forward? In our estimation, they come mostly from unmeasured confounding. And the unmeasured confounding, one way to try to understand that is to imagine, as it is very often the case in cancer, that there are subgroups that are different molecularly, that don't necessarily reflect, these differences are not necessarily reflected in age or stage, but they are born out of the fact that the molecular mechanism driving the cancer are different. And so there are these subgroups. These subgroups come with different gene expression patterns and with different relationship between the gene expression patterns and the outcome. Now, if these subgroups are represented differently across different studies, then what you observe at the study level is say, thinking about, in the simplest of cases, a coefficient relating one of the gene expression variable to survival. What you observe then is the result of merging across these different subtypes. And so it's some sort of a marginal effect. The more biologically correct effect would have to condition on the subgroups. If that conditioning doesn't take place, then and the mixing of different subgroup changes across study, that coefficient will change. And I think that's what's driving the result that you see in this specific case. It is something that is very, very important and generalizable. I mean, in cancer, the subgroups have a very specific biological foundation, but in general, we are very seldom to the point in health where we understand the heterogeneity of the patient populations to a level where we can get a complete full list of all the features that we need to characterize it. And it is also very seldom the case that as we switch from a population to another, those subgroups would remain unchanged in their proportion. So this is a danger that is far more general than just the gene expression one. Okay, with that, so that was just an attempt to document some of the sources of variation. It is very anecdotal in that it is for ovarian cancer or for breast cancer or for whatever other specific problem you have. It's not an attempt to paint a general picture. But I think it gives a blueprint for how, given a specific area of interest for AI in health, one can get a comprehensive view of many of the data sets available, as comprehensive as possible, and then use that to generate in silico data that would inform about the replicability and generally the performance of the algorithms, and giving confidence that applying them in a more general way would be efficacious. Okay, now, as for the implication for methodology, this is an anecdote again, but one that we found to be worth thinking about thinking about fairly seriously. So, you know, we established that cross-validation and cross-study validation, there's a gap between the two in quantitatively. And so in cross-validation, you tend to overestimate your ability to classify when you then move the model outside, well known. But is it the case, is that gap going to work similarly across a range of different ways of training the data or not? Obviously not. So if you are trying to choose between different modeling strategies based on cross-validation compared to cross-validation, you're going to end up with very, very different models. This is an example where we raised six common strategies. You see lasso ridge regression, as well as super PC and a version of boosting for survival data, univariate Cox, and this very, very simple algorithm called clipping, which is the brainchild of one of the greatest living statisticians, in my view, David Donahoe at Stanford. Clipping is something that he proved to be optimal for detecting faint coordinated signal amidst a lot of noise. And it is essentially a linear regression where you do a variable selection, and then coefficients are only allowed to be +1 or -1 after some standardization of the feature space. And this is something that we came to because the doctors came up with very similar algorithm in the early days of gene expression data analysis. And in fact, the first FDA approved algorithm, MammaPrint, was essentially using this, coming up with a list of good prognosis expression levels, bad prognosis expression levels, calculating the two averages and summing them up so that the negatives wouldn't cancel out the positive, and whence the -1. So that's essentially a way of reinventing the clipping algorithm. We codified it and studied some of the theoretical property, called it más o menos because it seemed like an ironic way to think about it, but then we discovered that Donahoe had arrived to that way before us. Bottom line is that clipping here does quite well in our in silico data-inspired experiments. It is when you look at cross validation, which is in gray here, it comes in the middle of the pack. But when you then look at cross-study validation, it is comparable. It is comparable to Ridge. Ridge is the best in cross-validation in this specific example, but it is comparable to clipping when you look cross-study. The algorithms that are far more complex, like Cox Boost and Super PC, don't do as well. And in general, we find that simpler algorithms that tend to get rewarded in terms of generalizability beyond the studies. And so that was motivation for trying to think about how can we learn the right degree of complexity by using a multiplicity of studies when we have the ability to do so in the hope of fostering generalizability. So yeah, so with that background, I want to tell you about some of the proposals to not only quantify these issues, but also to move beyond with the goal of engineering statistical learning methods that ultimately will validate well across samples. So this journey started more than 10 years ago, and is you know, our background is very much in terms of statistical learning more so than machine learning per se, although we're very interested and very open to trying various machine learning approaches. But most of what I will see applies to things that are at the interface between machine learning and stats. And the goal is in part to come up with good algorithms, but mostly to try to think a little bit more conceptually about what are some learning architectures that might work and might allow us to scale up relatively interpretable and simple methodologies to the multi-study setting. There's a lot of overlap between the things that I will present and work in meta-analysis, work in domain adaptation and domain generalization, multitask learning, and other areas of machine learning. The niche here, though, is, as I said, to try to stay simple and do something that is scalable and interpretable for health applications, for situations where simplicity is a way to retain some multi-study generalizability. I think I have a few examples of unsupervised learning and some examples of supervised learning before I get to the decision theoretic stuff at the end, and I hope to give you sort of a high-speed view of all of those going forward. Starting with unsupervised learning, this is a paper that we we published in 2018 with Roberta De Vito, who's now in data science at Brown and others, that generalizes factor analysis to the multi-study setting. And this generalization is one where you take case studies, each with its matrix of data and predictors that are fully aligned across the rows, and try to decompose each matrix into a standard factor decomposition where you have a small number of feature-specific coefficient vectors and a small number of individual-specific coefficient vector to give a low-rank representation of the data matrix, as I'm sure many of you are super familiar with. In this case, we generalized it by looking at doing the decomposition for the case studies jointly, and doing it so that there are some common factors represented at the bottom as the green and orange factors, as well as potentially some study-specific factors. The study-specific factor could represent special factors that are only present in a particular patient population or only detectable with a specific technology, but they could also represent things like batch effects or other artifacts that may be important to recognize and remove. So just to give you a quick motivation, these are two matrices from two matrices of factor loadings from the paper, and you can recognize that some of the factor loading look quite similar, and estimating them jointly would increase the efficiency. You can also see that some of the factor loadings in one of the matrices appear as one, and in the other appear as two somewhat highly correlated factors. And the joint approach will give you a way of robustifying the identifiability of that and coming up with a more sparse representation of the factors. And then there are some factors that are unique to these studies, to each of these studies, that could potentially be the result of batch effects. At the bottom, you see just the empirical correlations between the vector studies that are identified in the two. The next step beyond this, yeah, I don't want to dwell too much on it, but we have extensively explored the ability of these joint factorization to both reconstruct covariance matrices in the individual study at a higher degree of precision than doing the various decomposition one study at a time, and also in terms of prediction of rotating features out and trying to predict them based on the decomposition gives better results this way. On the other hand, merging the study and doing a single decomposition misses some of the study-specific features. So you could include, there are factor analysis methodologies that allow you to use covariates, but having the analysis be entirely stratified by study gives you much more flexibility and power than using the study label as a covariate. We generalize this to the situation where the factors are allowed to appear in any subset of the studies. In this case, for example, the blue factor now appears in 2 out of the 3, and we call that the combinatorial version of that, and Izzy Grabsky led that effort, which just came out in the Annals of Applied Statistics last year. And last but not least, we're spending quite a bit of time actually on negative matrix factorization type of decompositions these days, in part because of interest in mutational signatures in cancer, and going back to Dr. Calimeri's conversation earlier in collaboration with some of the colleagues in multiple myeloma, including the Munshi lab. So in cancer, there's quite a bit of interest in trying to understand the patterns in single base substitution that are found when the cancer is detected and sequenced. And these single base substitution are cataloged in an alphabet that usually comprises 96 possible categories. So the matrix M here represents a patient by category matrix, where it's a 96 by number of patients. And this is decomposed into a P matrix that has weight, that is vectors of weights on each of these categories that represent the signatures. For example, if a cancer has been exposed to smoking or a lot of sun exposure via UV radiation, it's only certain types of mutations that appear in a coordinated way in those cancers at a higher frequency than normal, and so you can detect it and tease out through this type of basis functions. And E represents the exposure of the individual. So it's individual level vectors that decompose the M into the contributions of the various different signatures. So it would essentially, in the intuitive example given earlier, it would quantify how much of the mutations of a certain types are due to sun exposure or smoking or other factors that are often related to endogenous things like the defects in fixing errors in the DNA when they come up. So there's quite a bit of biological background for this, but as a non-negative matrix factorization done on discrete data, it is a tough problem computationally and statistically, because it borders it's non-orthogonal decomposition, and it's very difficult to identify the individual components. So in this model, we do it on multiple studies to try to robustify the inference, and we also do it in a way that that's the subscript S, but we also write down the decomposition into two major parts. One that has an R superscript for recovery, where we have a very strong prior on P based on mutational catalogs and other previous studies that tell us what signatures we expect to see there. So the smoking signature, the sun exposure signature, are driven by very strong prior because we know how they should look like, more or less. The prior allows for a little bit of wiggle room, but essentially it anchors the signature to a certain region. And then there's a second part that is identical, but where the priors are a lot more vague, where we allow ourselves to discover novel signature in the data set that we're analyzing. The matrix A that you see in the middle is a matrix that drops signatures in and out of the study in a way that is very similar to what I showed in factor analysis before. I don't want to walk you through the notation, but that's a binary indicator of which signatures are part of which studies. Some will be in common, some will be only be in certain studies. And then on top of that, we add a covariate modeling where the exposures now are modeled as a function of covariates that are described by these XJS. They are specific to the study, and the individual with some coefficients delta S for this gamma regression that themselves have a mixed prior that allows us for some sparsity. It's a point and slab type of prior. So that's one of the direction we're going. This paper is being reviewed now. It's on the archives. We're hoping that Genobiology will like it. They like the first version of it, so we're keeping our finger crossed. But this gives you a sense for some of the application of this multi-study learning in the unsupervised way. And I put it in here in part because so much of the work that we do nowadays in health relies on representation-based learning, and these are sort of as complex as this has turned out to be at this point, a relatively more intuitive and biologically minded representation learning that can then be used for prognostication as well. So going to the supervised, I want to highlight mostly stacking-based approaches and an architecture that we've been playing around with for 4 or 5 years that goes back to that slide that I showed you at the beginning that showed the modest correlation between the alternative algorithm for ovarian cancer, and where I complained about the fact that we initially expected good success from ensembling, and we weren't able to go very far with it by simple ensembling approaches. So just to define the problem, we have multiple matrix of predictors corresponding to multiple studies, in this case, case studies. The predictors in all the illustrative example represent gene expression, but you can put in whatever you'd like. And then there are labels. In this case, the labels are binary, but they don't need to be. And the goals for the multi-study learning are what statisticians would describe as generalist or specialist, and maybe from a computer science standpoint, this is more like domain generalization versus domain adaptation. But you can think about having the case studies where you train, and then you are interested in an unseen domain for studies K+1, or in more patients of the same type for study 1. Studies in health could represent different health systems. They could represent different platforms for imaging or for measuring high throughput biological data. They could represent different electronic health systems that code data in different enough way to have a whole set of idiosyncratic divergences at the feature level and so forth. So those are the two main problems. We also often think about what happens if we know a lot about the target distribution, but we don't know the labels. So in this context, I want to describe a general architecture that we've played around with for training data sets in a way that we find to foster replicability across multiple studies. And it is based on ensembling. It is based on often ensembling using multiple learners. So we have multiple learners and multiple data sets. With multiple learners and multiple data sets, you can come up with a collection of algorithms. In this case, you have 6 3x2. And the goal then is to ensemble these with weights that reward the extent to which each of the algorithm, which initially has been trained on only one study, is good at predicting in the other study that are part of the overall training collection. So to reiterate this in terms of the matrices that I showed you a minute ago, it's a two-stage approach. We start out with the K-label studies. We have a collection of potential learners. For each of the learners, we train that learner on each of the case study. In this representation, just for simplicity, I have only one learner. So with that one learner trained on every study, I get K algorithm for making predictions. Now I can go back to each of the individual study and evaluate those K predictions on each of the patients in the case studies. And if I started out with the yellow and blue matrices at the top in terms of my features, I have now generated scores represented in red at the bottom for each patient using each of the K algorithms. Now, I'd like to point out that only a small fraction of these predictions are based on the same patients that were used to train the model initially. In fact, only one row for each of the K red matrices. So stage 1 is to separately train the learner, and now stage 2 is to merge the red matrices and the labels into one and train a learner to predict Y based on the red stuff. And that is essentially a stacking procedure, but it's a multi-study we call it a multi-study stacking because of this stratification by study. It has a small amount of data reuse, not a lot, and it mostly rewards rows that will do well even though they're trained in one study when they're faced with the merged data set. And this architecture, we tried many variations, but it is one that helps us making progress even in the very tough ovarian cancer data case, and it's been fun to play with and successful in many different applications. This is some work from the lab since the time we decided to focus sharply on that. ZOIG1 looked at the transition point for this type of architecture between merging the whole data set, the whole the K data sets, versus doing this type of stacking. And it turns out that at least in random effects models, there is a continuum in the trade-off between the model performance of the two approaches, where if the cross-study heterogeneity is small, you merge, and as the cross-study heterogeneity increases, at some point you cross a transition point where it becomes optimal to do the multi-study stacking. Katisher has done the same thing for boosting, generalizing the linear model results of ZOIG1. Maya Ramchandran did 2 papers in the context of random forest, and I'm going to say a little bit more about the second paper. I think I want to do that and skip the decision theoretic, more philosophical part. Maybe we'll talk about that offline. On an algorithm that we call cross-clustered weighted forest. I'll tell you all about that in a little bit. Gabe Lowinger devised a methodology for, given the collection of case studies, to create a continuum between merging and multi-study stacking by various clever ways of stratified resampling from each of the individual studies. Boyoran studied some of the theoretical properties and devised a sort of an optimization-based way of redefining the same stacking that also allows him to refine the approach so that there is no data reuse at the second stage. Gabe and an MIT student of Rahul Mazumdar named Kehan have done this sparse multi-study version of this that is going to hopefully appear in RSSB in the not too distant future. That is based on a discrete optimization version of the variable selection part. We are also venturing into causal inference, and Katisher has a paper on generalizing the R learner so that the propensity score part of the model is driven by multi-study replicability. And then the last line is for the decision theoretic replicability paper that I might or might not have time to cover. So 11:26, 4 minutes. I think the wisest thing is to just go over the cross cluster weighted forest. Before we do that, let me remind you where we are. So this architecture, which I explained it quickly, but I hope it's simple enough that you can grasp it. You first train K algorithm. You pick a trainer, train it separately on case subset, in this case the studies, and then at the second stage you merge and stack. This architecture, Maya had the intuition of applying it when you don't have case studies. You have a single study, but you cluster them and define the studies within the training set that you have to handle heterogeneity within the data collection in the feature space. So this is designed for situations where you have a lot of heterogeneity in the feature space, not much heterogeneity in terms of the concept shift or the relationship between the features and the outcome. And as a proof of principle, we started out with random forest. Random forest has been around the block a while, and it works really well in a lot of applications. And Maya's idea really blew my mind because it can improve on random forests really substantially in a lot of applications. And I'll show you a simple simulation example. Essentially, the algorithm is the algorithm that I described, except that you pick on top of picking, you choose the random forest as your learner, and then you pick your favorite clustering algorithm. And we played around with quite a few, and I think what I'll show is just some simple k-means, but there are many others that you can use to define the studies instead of having the study come from some meta-analysis. So I'm going to show you the results of a quick simulation where we have 5 studies for training and 5 studies for testing. The generative model is roughly linear. The coefficient for the variables are either 0, -5, or they live in these intervals that are bounded away from 0. You go from -5 to -0.5 or 0.5 to 5. Random noise and some variations with non-linear combination of the covariates as well. So this figure summarizes the results, and I hope to be able to walk you through what some of these lines are. The x coordinate refers to the number of clusters that we decide to partition our data set on. The vertical scale is the average RMSE over a bunch of simulation, and there are obviously versions where we show the variability as well, but this is more concise as a way to tell the story. At the top, you have random forest on the merged data set, which is the standard strategy that we might apply to this data set. Then going down, we have the flat ones are the strategies that are based on merging, and they don't depend on the number of clusters that you pick. And we fit a neural net on the merge, which in this case does quite a bit better than random forest. We fit a multi-study learner that is based on the previous architecture and knows the correct clusters that were used for generating the data, because the data is generated, the generating model here is generating based on known clusters. So the green is a little bit of an oracle because it knows the correct number of clusters. The clusters, however, are quite overlapping, and it's not necessarily the right way to partition the data if you're going to do random forest. And then the red one is the multi-study again, but where the single study learner is the neural net. Then the lines that depend on the clustering are the cross-cluster weighted forest. And if you randomly partition in cluster, you get the black curve, which already does quite a bit better than some of the algorithms that don't cluster. But if you cluster even remotely wisely, which I think we use k-means in this case, you get these two descending curves, the purple and the blue, which can do quite a bit better than many of the other approaches. The gap between the sort of default random forest and some of these algorithms is quite substantial, which suggests that there is something interesting about these architectures that is worth exploring further. We have now gotten some interesting theoretical results based on linear approximations of the random forest that we are hoping to submit very soon, and they are in the archived version of this paper. Okay, 1131. I think this is a good point to give you sort of maybe a bit of a lesson learned to wrap up. My philosophy in general is that these science of data science exercises where you grab very large amounts of data as comprehensively as possible and try to empirically analyze the performance of statistical or machine learning methods in a systematic way are really fun to do and useful. And if that's your perspective, you're going to have to deal with multiple studies, and that begs the question of how do we learn how to do well on multiple studies. So I think there is value in using multiple studies in statistical learning, and there's potential to have that to be a big driver of how we improve the generalizability of the algorithms that we use when we are going back to the clinic and trying to try them out in new hospitals, new imagers, new health systems, and so forth. Much remains to be investigated from a theoretical point of view as well, and so that's also something I hope more people will focus on. And with that, I thank you for your attention so far, and I'm very, very happy to try to answer any questions you might have.